In this video tutorial, we use cBioPortal to explore key insights into a specific type of cancer, which shows lung adenocarcinoma due to its high prevalence and mortality. Lung cancer is the leading cause of cancer mortality worldwide among both sexes and men and women separately. And lung adenocarcinoma is the most common subtype of non-small cell lung cancer. Also, it has diagnostic and therapeutic challenges that make understanding molecular mechanisms important for the development of more effective therapies. And it has a huge impact on public health. We can query lung adenocarcinoma in cBioPortal, and in this case, for our resolution study, we decided to choose the Pan-Cancer Atlas as it provides the most recent genomic data, including very important information such as mutations, methylation, messenger RNA expression, copy number alterations, and clinical data for each subject. Recent literature, such as the publication you can see in here, states that women might be more susceptible to lung cancer and indicates that there is a higher rate of lung cancer in non-smoking women compared to non-smoking men. This might be due to the fact that women tend to have other predisposing mutations. And we want to continue a further analysis on the overall genetic alterations in lung adenocarcinoma and how they differ between males and females.
We selected 3 genes for further analysis. We chose the first most frequently mutated gene TP53 in lung adenocarcinoma for further analysis, and 2 other genes which are well characterized and had either gain of function or loss of function mutations to show as examples. These other 2 genes were KRAS and KEAP1. Both have been described in previous studies of lung cancer. We can type in the gene names at the top right search box and press query to look at individual genes. Clicking the mutation tab on the top menu allows us to see a lollipop plot showing the sites of mutations in each gene. Each lollipop represents a mutation, and the height of the stick indicates the number of patients that have the same mutation at the same time. Domains of the protein are colored and labeled, allowing us to see where mutations cluster. Hovering the pointer over the blue circle under the annotation column provides more information on the gene, specific type of mutation, and biological effect. There is also a fire icon which indicates a recurrent hotspot. It is also worth mentioning that you can add OncoKB and cancer hotspot tracks to see whether the mutations are common driver mutations. As a gain of function example, we can look at KRAS. KRAS carries a gain of function mutation, and the lollipop plot shows a hotspot region where a G2C mutation occurs, and it is observed in 152 patients. This mutation is marked as a recurrent hotspot, which indicates that it is likely a driver mutation, and the blue icon shows that it is a gain-of-function mutation. This mutation occurs at a frequency of 29.9% in this specific study. For the loss of function example, we can go back to TP53 example. This gene has the highest frequency of mutations, which is 52.1%, and this gene usually undergoes a loss of function mutations, which result in a loss of tumor suppressor activity. This is confirmed by the blue icon, which indicates a loss of function mutation, and a fire icon, which indicates a recurrent hotspot. As you can see, there are many recurrent hotspots and many OncoB annotated mutations. Unlike KRAS, TP53 has more hotspot regions, and many of which are driver mutations. KEAP1 is another example for loss of function mutation. This gene has one more significant peak, an amino acid change, a G change to either R, E, V, or W. We can press on the lollipop to see all 5 mutations. However, as you can see, there is only 5 patients in this peak, which is not as significant as in the case of KRAS. However, the mutation is indicated as a cancer hotspot, which indicates that it is possible that it's a driver mutation. Overall, a lollipop plot is a useful tool to visualize hotspot regions and compare them to information from cancer databases.
So to generate the oncoprint, we just need to find our study. In this case, it's lung adenocarcinoma, PanCan, and then yes, we select the study, we query by gene, we type the genes of interest. In this case, they are TP53, key EAP1, and CRAS. When this turns green, we can submit. Okay, so every time that we query by gene, we start on oncoprint. As we can see, oncoprint is a nice summary of all the data, and it is important to mention that we can zoom in and play around, zoom out to see the data more clearly. Every column here is a patient and every row is a gene, and it is colored according to the types of alterations. The colors are defined below. So there are a lot of mutations in TP53. 52% of the samples have a mutation in TP53, then 20% of the samples have an alteration in the second gene, KEAP1, and 33% in CRAS. There is a mix of mutations, as it can be seen. There are deep deletions over here, where they are represented by the blue color. There are truncating mutations represented in black. There are splice mutations in orange. There are missense mutations. There are amplifications. Interestingly, another feature of oncoprint is that different tracks can be added to visualize additional information, such as subtype, for example. And then we can see that all of them are lung adenocarcinoma. Then, I don't know, diagnosis, age could also be added. We can see first patient was diagnosed at 69, second patient 60, 47, and so on. Then we could also add sex track, and then we can see if the patient is a female or male. And again, if we want to know more information about a specific track or anything, we can just hold on the cursor on top to display this definition or the explanation. Additionally, we can choose whether we want to view sample or patient information, events per sample or per patient. In this study is the same, there is only one sample per patient, so it is the same. This plot is also downloadable. When we hover over a mutation, we can see this specific mutation. We can see that it is statistically a hotspot and that it is a likely oncogenic mutation. We also have the link to the patient, and here if we press, it will show us all the specific information of that individual. And that's it. This is OncoPrint.
I will now go over how we can look at gene expression using box plots on cBioPortal. So first of all, we select the study that we are interested in, which is this one in our case. And then since we are going to be looking at the expression of a specific gene, we go into query by gene. And then in this box here, we can enter the name of the gene that we are interested in. So in this case, I will choose the gene KEAP1 because as mentioned earlier, it has an interesting hotspot mutation. So it would be interesting to study it, and it is also maybe a less known one compared to P53, so we thought it would be good to have a look at it. So I enter the gene name here. Once this turns green, I can submit query. And then when this is loaded, you go into the plots section here, because this is where all the plots showing gene expression of the specific gene will be. Now there are several different things that we can look at. First thing that we can see, for example, is looking at mRNA expression levels based on, depending on mutation type. So down here you've got mRNA expression, that's already selected, but up here we can change this to mutation, and then this will show us the different types of mutations here and mRNA expression here. So we can analyze these plots. So as we can see on this side, this is mRNA expression levels for no mutations, which is about 1.5K. And then missense, for example, seems to have higher mRNA expression levels, whereas truncating mutations have lower levels. So that's how we can analyze this plot. Then another thing we can look at is mRNA expression levels based on what type of copy number alteration the gene has. So instead of selecting mutation here, we can select copy number, which will load a new plot. Then if you scroll down, you can see the different types of copy number alterations here and mRNA expression levels here. Now, as we can see, this is quite logical because deletions have lower expression levels compared to diploid and gain copy number alterations, which makes sense. And then lastly, we can look at our biological question, which is related to how mutations differ between males and females. So you can compare expression levels between men and women. If here you could select clinical attributes and then sex as the clinical attribute that we want to look at. So we can compare mRNA levels for women versus for men, and as we can see, they seem to be quite equivalent. So these are just a few example plots of how you can look at gene expression on cBioPortal. There are other things you can select here as well, like structural variants, methylation, etc. But overall, this is how you generate gene expression plots in cBioPortal.
Now we're going to talk about how we can analyze mutual exclusivity of DNA alterations in the cBioPortal. So first of all, what is mutual exclusivity? So basically, it is a pattern that is observed in cancer genomes where specific genetic alterations or mutations tend to occur independently of each other within the same set of tumors. So this basically means that if one specific alteration is present in a given tumor, the likelihood of another specific alteration being present is significantly reduced. So now we are back at the cBioPortal, and we're going to now search for our study again, which was lung adenocarcinoma. And it was this one here specifically. What we're going to do now is use the query by gene function, and we're going to add the genes that we're interested in. For the sake of simplicity, we're just going to stick with the ones that we already used in our previous studies, or like in the previous tutorials, which were TP53, cAB1, and KRAS. And then we're just going to press on Submit Query, and we are going to obtain the following. So looking at this plot, we can already get a feeling of the data and make some guesses about the co-occurrence or mutual exclusivity in the different samples. Just so you remember, we can zoom into the figure, and then it's easier to see that actually each of these columns represents one patient. For some examples, we can first look at the TP53. For example, in this area, we would have, or it seems like we would have, a mutual exclusivity, while, for example, in this little area, there's not a mutual exclusivity of TP53, because we also can see that KRAS can be found in the same patient. However, we want to have more detailed information, especially also statistically significant information, and that's why we can do the following, which is going and click on a mutual exclusivity tab over here. So in the first line of the table, you can see the comparison of the KRAS mutations and the TP53 mutations. Here you can see that KRAS and TP53 are actually considered significantly mutual exclusive, because the p-value and the q-value are both low. That's why we can say it is significant. Furthermore, we can see that we have a negative log2 odds ratio, and this shows that the presence of TP53 is more likely than the presence of the KRAS mutation. In the second line, we are comparing TP53 and cAB1. They also seem to be mutually exclusive, but when we check the p and q values, we can see that this result does not seem to be significant. Here we also have a negative log2 ratio, so we can also say that the presence of cAB1 may be more likely than the presence of the TP53. However, again, this is not a significant result. And in the third line, we have a look at KRAS and cAB1. And actually what we can see is that they are not mutually exclusive, but they may be co-occurrent. And the positive log2 odds ratio here indicates that the presence of KRAS may be more likely. But again, this is not a significant result because both p and q values are too big. If you look at the table here, and if you decide you want to only display mutual exclusivity, you could, for example, unclick or reselect everything that interests you. And same here for the columns, you can add what you want to see. Lastly, if you decide that you want to have a look at different genes and different mutations, or add some more, you can go to modify query, or, which is a bit quicker if you only want to add some more genes, you can go to this pen and just add whatever you want. Let's just say TTN. We submit the query, and then you can see that TTN gets included into our analysis.